Hello, everyone. Welcome, and thanks for joining us today for this AGC Biologics webinar. My name is Ellen Chichevsky, and I'm from Samba Scientific, and I'll be the moderator for the webinar today. One of the most critically underdeveloped aspects of industrialized cell and gene therapy manufacturing is rigorous regulatory strategy and process design. Today, we're joined by two experts in this field who have recently been making waves across the globe, sharing their insights and analysis. Our speakers are based in Milan, Italy, at the AGC Biologics Cell and Gene Center of Excellence. We have Monica Pema, Senior Manager of Manufacturing Science and Technology, and Francesca Rossetti, Analytical Method Development Director. The title of today's talk is Uncharted Paths, Successfully Validating Your LV Gene Therapy Product Process to Gain FDA and EMA Approval. We look forward to hearing from Monica and Francesca about their experience of guiding an LV therapy to market.
Thank you, Ellen, for introducing us, and thank you everyone for joining us today during this webinar. Myself, Monica, and Francesca are honored to present on behalf of AGC Biologics our experience with lentiviral vector manufacturing and testing from early development and clinical phases until commercial phases. So few words about AGC Biologics and what we do. We are a wide world company since we have 8 facilities and more than 2,500 employees distributed through Europe, US, and Japan. The two of us, as Ellen anticipated, work in the Milan site located in Italy, which is a site that has been assigned as Center of Excellence for Cell and Gene Therapies, since we have obtained regulatory approval from different agencies. The latest one is by FDA, and we have proven technical and scientific experience in the manufacturing of lentiviral, retroviral, and AAV vectors in both adherent and suspension platforms. Importantly, we have experience and strong proven experience with the manufacturing of different cell therapies, including CD34 hematopoietic stem cells, drug products, T cells, NK cells, human mesenchymal cells, and generally speaking, different cell types. In particular, we have a wide spectrum of analytical methods that we can offer internally, performed in-house to all our clients to support their activities. Outside of Milan, you can find these similar services also in our twin facility located in Colorado, in Longmont, in which we have transferred the expertise so far built in Milan, also there to expand our services for cell and gene therapies. In this slide, you can see an overall picture of the advanced therapies that we have provided so far as AGC Biologics, but today's focus is about the process validation activities that we have performed for our clients. And in the next slide, you can see the state of art of in vivo and ex vivo cell and gene therapy products that have been approved by either FDA and or EMA. On the right side, you can see the ex vivo therapies, and you can appreciate that among 17 ex vivo cell and gene therapies, 6 of those, which are highlighted in green, are manufactured in the Milan site of AGC Biologics. We have 3 approved products by FDA and or EMA, and another 3 cell and gene programs that have been submitted to for approval, but we are still pending the approval. And we anticipate that a couple of more products will be filed in the next 2 years. So how AGC was able to be so present in the ex vivo cell and gene therapy field? The answer is simple. We worked very hard together with our clients and supported them in achieving their successful process validation activities. In this slide, you can see the production of our process validation work in the past 10 years. As you can appreciate, we have manufactured 12 successful PPQ programs, 4 for cell products and 8 for vector products. And we have served, in general, 5 clients, but we have also worked strongly on one proprietary program. Since our work does not finish with the successful PPQ, we continue supporting our clients also in post-approval change management activities, and we are currently having a series of activities for our approved RVV, LVV, and CD34 cells drug product. So one of the questions we got asked frequently is how we do process validation for cell and gene therapies, and the answer is quite simple, since we apply standard 3 stages approach as per current guidelines. We start with stage 1, which is the process design, and at this stage we aim at building up and understanding our process by identifying CQAs and CPPs, and we focus on in-depth process characterization studies to expand the knowledge of our process. We basically build up the process to bring forward to GMP manufacturing for the initial clinical trials, and also the first draft, let's say, of the control strategy. During the following stage, the stage 2 of process qualification, what we do is to evaluate if the process designed in the first stage together with the control strategy are capable of performing successful manufacturing process applicable to commercial future applications. During this stage, we also verify that the qualification of the facility, the utilities, the equipment, and also our personnel are performing as per the intended commercial process. Finally, during the stage 3, we work on the continued process verification, during which we ensure constant and continuous monitoring that our process performs as expected and is maintained in a state of control. There is one important point that we should never forget about, that when we think of the process validation, we have to think also about the analytical methods, since the analytical methods part is something that follows each of the stages of the process validation and gets more mature when we move from the early beginning to the stage 3 activities. But I will not go into details about this, since Francesca will speak more about methods later on. So in the following slides, I will show you the activities that we have performed during the process validation, and in particular I will present you a few examples of a case study for the stage 1 activities that we have performed for one of the lentiviral vectors that we manufactured at our facility. The slides are not scrolling very well. Apologize for that. They should. Okay, no, sorry. Yeah, apologies. Yeah, I truly believe that the stage 1, which is the starting point, should be empowered as much as possible, since we start understanding and start building everything here. Stage 1 starts in process development. We start developing and optimizing the process and characterizing it, and we also transfer it to GMP for clinical production, while in parallel we work also on the analytical methods, and we develop and qualify product-specific analytical methods, while for the remaining part of the analytical methods that are used to test the products, we can also rely on the platform methods that are available at our CDMO. So to to characterize and to understand the process, we have an important element in our hands, which is the scale down models. We have scale down models that are mini versions of the manufacturing process, in which each parameter and equipment has been mathematically scaled down in order to be full representative of the full scale process. So these are very reliable tools for the characterization of the process, and not only, we can use those also to test, for example, new genes of interest, and it happens very frequently that our clients start with working with one lentiviral vector, but then wanted to use the same manufacturing process to test different versions of other lentiviral vectors, and we use always the scale down model, the scale down models, which are indeed very supportive, because are considered a limited economic investment that can provide results in very quick timelines and expedited timelines. So here at AGC, we have different scale down models for vector production, both for the upstream. You can see in these slides are the models that we have for the upstream phase, for the cell culture. And in reality, the platform process that we offer as a CDMO can be scalable both upstream and downstream, and different parameters are scalable. What you see here in the slide in the red circle is shown the scale down model that we have used for the execution of the process characterization studies that I will show you later during my presentation. In the second slide, okay, here are the scale down models for the downstream phase. We have focused mostly on the chromatography and on the concentration steps of our process. So, okay. So I told you about the importance of characterizing the process and about having the scale down models that are representative of the process to accelerate your process understanding. But everything starts with the analysis of our process parameters. So we do a risk assessment of our process, and we identify the impact of that the parameters have on the quality attributes of the vector, and we assign criticality risk to these parameters, and depending on that, we decide the asset of parameters that need to be studied in depth with specific process characterization. So once we have the tools in our hands, we then decide which of the parameters are requiring in-depth studies. Going to the the case study I was mentioning before, I will now tell you a little bit more about a set of process characterizations that we did for 48 liters scale lentiviral vector manufactured in addition in factories. At the beginning, the first PC study that we did is about the seeding density at the inoculum step. For those of you who are familiar with this type of process, you must know that this is a very particular step in which we seed the cells, and we know that the appropriate confluence of the cells at the inoculum step is crucial for a very productive transduction and efficient step, and the good production of the lentiviral vector. Another example of a process characterization study that we have done is about the DNA transfection step, in which we have characterized in depth the transfection mix. We have studied the different amount of plasmids that are present in the mix, and the amount of the transfectant reagent that we use, and the ratios between all of those. Moreover, for the transfection step, since it is crucial to the production of the lentiviral vector, we have also studied the incubation time with cells, for example. Following this set of studies for the upstream, we have also studied in depth the downstream process, and we have concentrated and focused on the chromatography steps, the anion exchange, and the size exclusion chromatography. For these two steps, for example, we have studied the column geometry parameters for the packing criteria for columns, but we have also studied the elution selectivity and analyzed the eluted peak in order to determine the most appropriate parameters to be used in the process. And in general, for the chromatography steps, we have also studied in depth the load ratio by challenging the performance of the column to understand the limits for the usage in the process. The process has different filtration steps. We have the clarification step, but also the final sterile filtration. And in general, for the filters, the type of study that we do is again load challenge in order to understand if the dimension, the surface, but also if the type of membranes are appropriate for the intended use in the process, and if they are retaining the impurities in the appropriate way. For the filling, for this scale of process, the filling is manual, so we don't have that much PC studies in design, but what we do is to study for example, the fill homogeneity of the manual filling. We also have studied the vector freezing profile, and we have also studied the stability of the vector, both at long-term, accelerated, or intermediate, and stress. So all these elements that I just showed you are very helpful to build up very most complete control strategy. But we are aware that not every time you can have the full elements that I just showed you, and you cannot have a huge amount of process characterization for different reasons. So when you define your control strategy, since this is the goal of stage 1, there are some critical aspects that you should consider in the definition of your control strategy, and you should consider in order to be prepared on what to expect from your control strategy, and also to anticipate what will be the next requirements for the next steps of your process validation journey. For the control strategy of stage 1, you should consider a critical element of the potential reduced number of batches available at scale and or scale down, because you might have been doing a few number of batches during the development. You should also consider, since it is critical, a limited analytical testing data set, which might result then in a difficulties to set acceptance criteria for all of your tested CQAs. When our clients decide to use our platform processes, we have a series of studies already performed to characterize the process, and those are provided together with the platform process, but you might be aware that this might not be sufficient, and each client might have different needs, so you might need to also design and execute different additional characterization studies to feed your product specific needs. So these are all elements to consider, because all together help you building the control strategy. Another element that should be considered is the limited budget and tight timelines, which are usually dependent on the client. So that's not that much we can do on that as a CDMO, but for sure we can help you do the best we can with what we have, the materials that we have in our hands. So moving to the to the stage 2 or the process qualification activities. One of the most frequent questions that we receive as a CDMO is how many batches, how many PPQ batches do I need? Unfortunately, there isn't one number that fits all the situations. Because the PPQ, for the definition of the PPQ batches, a justification exercise is required. The PPQ number of batches can go from one single batch. This is very hard to be explained and justified, and honestly, we haven't experienced that, and the other batches can be done in concurrent validation to 6 to 8 batches, which is more common for the validation, the qualification of the cell processes, which are exercises that are done using healthy donor material. So the cells are very limited, and you have also to do studies on the stability of drug products. So this is why you have to increase the number of PPQ batches that you do. Usually, in our experience, we have manufactured at least 3 PPQ batches as a standard. As a standard approach. Regarding the PPQ readiness, what else we should consider is the important part is to arrive at the stage 2 at the PPQ with well completed stage 1 activities and completed on time, due time for the PPQ readiness. This will give you a good process knowledge, a good set of data, and will support you building up the justification for the definition of the PPQ batches needed. Moreover, you have also to consider generating a solid set of data from analytics, since this will support you defining numerical acceptance criteria for all the CQAs, and also the experience during the process understanding and knowledge will help you arriving at PPQ with acceptable ranges defined for your process parameters. I'm suggesting this all these topics since it is not always the case. Sometimes at the PPQ stage, not all these elements are in place, and it becomes more difficult then to put together and to have a very good PPQ readiness. So the bottom line is that we should make sure that the control strategy from the stage 1 and from the clinical phases is sufficiently comprehensive also to face the commercial phase, in order that when we arrive at the PPQ, we have to think also about minor revisions, few revisions, and few changes to the control strategy. So can you see the next slide? No, sorry. Now. Okay. Okay, so when you arrive at the PPQ and we concluded, we usually feel relaxed, since it is a very great achievement, but we have not to forget about the stage 3 activities and the continued process verification. Some must know that I can share with you, according to our experience about the CPV, is that this is a clear strategy that should be defined by the client, who is the owner of the regulatory strategy, and in cooperation with the CDMO, since whatever is in the client's strategy for CPV should be also supported by the CDMO. So we should make sure that what you want to monitor in the future, we are able to support you, and is information that we are able to continue providing to the clients. When you speak about the CPV for cell and gene therapies, we have to be prepared to deal in some cases with a limit number of commercial batches and data sets. since the demand for cell and gene therapies, it's not so huge, so they are rare diseases. So you have to be prepared about that, and you have also to think about identifying an analytical testing to be used during the CPV that it's informative and gives you an idea on how your process is performing during this stage. Moreover, the CPV needs a statistical analysis of your data and your data trending, and I understand that this is not always easy to set up, so you have to think very well about the statistical approach that suits better for each phase of your CPV review period, and that fits very well with your numbers, whatever those are. And for cell and gene therapies, again, very depending on the number of batches that are manufactured for the specific program, the CPV review periods can be very diluted over time. So the trending of your data can be very spread over time. So the very last slide, and I will go very quickly to this since we have here Francesca that is waiting to speak about analyticals. I think that the process validation is a very amazing journey, but you have to consider working through it, keeping very clear in mind which are the challenges that we should face together as in the relationship between the CDMO and the client. The challenges are multiple, so we have variability and of the starting materials. A variability of the starting materials can be sometimes challenging. The process is manual for the majority of the unit operations, so the variability of the process is high. We have high analytical diversity and analytical methods variability to deal with. The potency assays are not the easiest one to develop, but we have to work on those and to have to work as soon as possible. And as I mentioned lately, limited number of batches are a challenge, so we have to be prepared to work with them. And again, the statistical models, I mentioned those for the CPV, but it's not only the only part where the statistical models are used. We need statistical models for the analysis of the process characterization result. We need statistical models for defining the acceptance criteria. So altogether, this is the bucket of things and of elements that you have to consider when you think about your process validation journey. And now I think that it's time to go to Francesca's for her analytical part.
Hello to everyone. Monica has just nicely shown you the approach to the process validation for the cell and gene therapy product. And I want to show you how to bring analytical methods from development to the market. As for the process, it's very important that you start to think and to define your analytical strategy as soon as possible. To think which method you want to apply for the characterization of your product, which are going to be maintained for the release, which are the stability indicator method, and how you want to report your results. Just to underline you the importance of the analytical methods, we have just been approved by FDA, and 80% of the questions that arrived on the regulatory document from the FDA were questions on the analytical part. The questions range from method development to method validation, and they really went into the details of the method, spanning from scientific rationale, history of the method, and compliance with guidelines. And they requested the product-specific validation for each single method. The quality control and the analytic is very central in the process of gene therapy, starting from the bone marrow or the apheresis, the selected cells, the vector used to genetically modify the cells after transduction, The transduced cells in formulation medium and the reagent are all controlled by quality control. You need to think that in order to characterize or release a vector, we run about 25 analytical methods, while for the cell product, we run between 10, 13 assays according to the different project. So the journey, the work in order to have the analytics ready for the market is very long and complex. To go with the process development, we need, first of all, it is essential to have an exhaustive panel of analytical methods and high throughput testing. You want to have a robust inline process analytics to support the process development and monitor GMP manufacturing, and a release strategy to optimize the sample volume dedicated to QC and time to release. The status of the analytical methods, scientifically sound, qualified, or validated, is in accordance to the clinical phase and to the type of method. When we are in development and during the process characterization study, the method that you will apply for the characterization, stability, and in-process control can be scientifically sound, and safety methods are not mandatory. When we go into the clinical phases, the safety method need to be full validated, while the other method can remain scientifically sound or qualified. Where scientifically found, I mean that in my method I have all the controls to monitor the performance of the method and to give results that can be comparable among the different batches. When you arrive in the late clinical phases and in the marketed application, for sure all your methods need to be full validated, except for the method applied for characterization, which can remain in a scientifically sound status. The qualification and the validation of the analytical method according to ICH Q2 R2, this is the new revision that is going to be issued tomorrow. It depends on the type of method. So if I want to validate an identity test, I will need to study the specificity as parameters. Then I have the purity test that can be divided between quantitative and qualitative. For the quantitative methods, I need to verify all the different parameters described in ICH guideline, while for the purity test, I need to verify just specificity and the detection limit. For the potency test, for sure, I need to verify all the different parameters except the detection limit. In any case, if you are not able to verify one of the parameters because it's not possible, it is very important that you described in the document why you are not going to verify that parameters. For example, sometimes it's not possible to verify accuracy because, for example, there is not a reference standard. You can infer the accuracy by the linearity and precision, and you can explain why you didn't reach that parameter that it is described in the guideline. On top of this, I put the analytical challenge. First of all, the guideline. Most of the methods are not compendial, so they are not described in the different pharmacopoeias. We don't have specific guideline. We do have the guideline for the validation and qualification, but you will not find any guideline that tells you that, for example, for an infectious viral titer method, you may run a digital or a real-time qPCR, or that tells you which is the best identity method for your product. And we don't have guidelines that tells you the specification, above all for the impurities. We have a high variability. A lot of times assays are coming from the R&D, so they are not ready for the quality control without optimization. We have complex biological procedure, long time of execution, high analytical diversity, and absence of reference. And then we have reagent instrument. A lot of times we need to use research-grade reagents, and when we are in development or in qualification, we have one single lot available. So we don't really know when we are going to have the next lot, if it's going to work correctly and in the same way. Sometimes reagent changes. It happens, for example, with antibodies for ELISA. And then we have the instrument. Even if in the last years, the suppliers put a lot of effort in order to have instrument compliance to GMP, still there's sometimes we are in research, and for example, we do have instruments that are not, for example, compliance to CFR 21 part 11. How we can help in AGC Biologics? In Milan site, we are about 300 employees, and you need to think that 1/4 of the heads are dedicated to analytics. We have two teams, the analytical development and the quality control. Analytical development with 23 scientists, 48 scientists in quality control. High specialized personnel, we perform continuous training. We have different skills in molecular, cellular, and biochemistry. We run more than 160 assays in-house. We prefer to run assays in-house because we have more control of the methods, and above all, we can optimize the volume dedicated to quality control and the time of release. We are full compliance with GMP. We can give CMC support. We have a unit dedicated to microbiology and a unit dedicated to the automation. We have different capabilities. We can span from the molecular biology. We can perform real-time qPCR, digital PCR. We have different plate readers, plate washer. We can perform Southern and Western blot, chromatography. We have all the instruments to perform all the chemical, physical characteristics of the product, flow cytometry, dynamic light scattering, MFI for the subvisible particle, microfluidic technology, and the liquid inspection viewer. This is our platform. We set different platforms for the characterization of the different type of vector, adeno-associated, lentiviral vector, and retroviral vector. We do have a potency identity method, safety method, purity with a different residual proteins, residual DNA, and the residual of the reagents that are coming from the GMP manufacturing, and the physical and chemical characteristics. As I told you, 95 of these methods are run in-house for the motivation that I listed before, and we really outsource a few methods. We can also internalize a different kind of method and the product-specific one. We have a dedicated experience in design the potency method, design, develop, and qualify the most appropriate potency method with different readout. We can have transduction efficiency, flow cytometry assay, cytotoxicity, cytokine release, enzymatic activity, immunoassay, gene and protein expression. In this moment, we do run methods in phases clinical 1, 2, and 3, and at the market application. Just to give you a little bit of an idea of the work that you need to perform to set a potency method. For example, I don't know, for a CAR-T or a TCR. First of all, you need to select the cell banks, the reference and the target cell line. After the identification of the target cell line, you need to set the optimal culture condition. And then usually we produce 2 cell banks, the first stock and the second stock. The second stock is the working cell bank. After the cell bank, you need to define the experimental condition, the procedure of your method. So you need to set in the optimal culture condition and the transduction process. You need to identify the optimal effector target ratio. And for example, if you want to see a transduction efficiency, you need to set the panel for the flow cytometry. At the end of the setting and of the development of the method, we will perform the qualification according to ICH guidelines. Then, for example, you want to measure the production of cytokines in cell supernatant, so we can develop an ELISA test, for example, and then we can set the condition, the procedure, and qualify the method. You need to think that going from the development to validation of a potency method, it can take about 9-12 months. So it is something that you really need to think as soon as possible. We do have a precise analytical methods life cycle. Starting from the development of the method, we can develop the method, or the client can transfer the method to us. Then we have the qualification with the development and qualification report. Then we define the transfer master plan, and the method is going to be transferred to quality control. We will have the transfer protocol, transfer exercise, and the transfer report. At the end of the transfer, quality control will issue the method, will code it the method, and then when it is necessary, we do have the validation. And we will have the validation protocol, validation exercise, and the validation report. To give you a little bit more details, we start from the development of the method. As I told you, we are able to develop the method from scratch, or in a lot of cases, client is transfer the method to us. Usually the client transfer the method to the analytical development. We optimize the method in order to be fully compliant to GMP, and then we qualified and we transfer to quality control. Tomorrow, we are going to have the issuing of the new analytical procedure, the ICH Q14, that it is very interesting, and I invite you to read it very carefully. Then we have the qualification and the validation. In the qualification, we set the criteria of acceptability of the different parameters described in ICH guideline. So we define the criteria of acceptability for the accuracy, the precision, the linearity, the range. And then in the validation, we are going to verify the criteria of acceptability that were set during the qualification exercise. So, for example, during the qualification, we are going to study the precision of our analytical method. We are going to define that the methods present a precision and interest variability of 20%. When the method is going to be validated, the criteria of acceptability for the precision will be 20%, and it will be validated in quality control. I put a little bit of insight about the product-specific validation. This parameter was requested strongly by FDA. And it was not only the precision, not only the running of the different samples, IPC and final product in the method, but also to perform the spiking study, inoculating different concentration of the standard into the sample in order to define accuracy and linearity. You need to consider that it's not always so simple, because the vector presents inside some amount of some of the impurities. For example, if you want to define the accuracy, spiking different quantity of the plasmid DNA inside the vector, you need to consider that the vector presents a background, a certain quantity, and then you need to face this criticality when you perform these studies. At the end, we brought some of our analytics into the automatic station, and this was very important during the process characterization studies, where we needed to analyze a lot of samples in a short period of time. We brought the manual methods into the automatic station. We have a different automatic station with different liquid handling. We have an automatic station for the vector dilution for the cell transduction, an automatic station for the ELISA assays, and two different automatic stations to perform real-time qPCR. All the stations present a core liquid handling that is able to dilute sample, dilute the standard, prepare the plate, and then the ELISA station has integrated the plate reader and the plate washer, and it was decided in order to perform different ELISA analyzing starting from the same vial. So you can schedule different ELISA tests, for example, for the impurities, starting from the same value. And there is a software that is going to schedule the different procedure in order to have the results of different ELISA in the same day. For the automatic station of the qPCR, we have integrated the sealer and the instrument to perform the real-time qPCR, and it was designed a little bit different. It's going to run the same analytical method, the same qPCR a lot of times in order to really increase the throughputs. And these were very, very helpful during the process characterization studies. Just to complete, the process validation is complex, but your journey can be different. You need to invest to empower your stage 1 activities, to plan on time your stage 2 and stage 3. You need to take advantage of the knowledge of data available for platform process approach, continuous support from analytics, platform method, and in-house testing preferred, and strong scientific and statistical justification of the specification set. Thank you very much for your attention.
Thank you. Thank you to both our speakers. I appreciate your candid review, and I think there's a lot to learn from your experience in developing these methods. We have just a little bit of time for a few questions, so I'd like to spend some time on that. Please remember to post your questions in the Q&A tab at the bottom of your browser. I'll begin with a question that speaks to your sort of unique perspective in being familiar with EMA and FDA processes. We have a hypothetical scenario here. If a client is located in Europe and is performing a phase 1 clinical study at one of AGC Biologics' US sites, does the US site need to be registered to EMA? And again, it's for a phase 1 study.
I think that, we have to be very precise in answering about regulatory inspections, etc. We should maybe take some time to speak also with our quality group and regulatory group, but considering it is a phase 1, I think that if the manufacturing is performed in one of the US sites and the clinical trial is done in the US sites, no, the client who is the sponsor of the project does not need to be registered to EMA.
Understood. It probably varies, depending on the scenario. My next question is for Francesca. Do these validation and analytical methods work for all vector types, or are you developing methods specific to each vector and therapy that you work on?
So as I was showing, we set a different analytical platform for the different kind of vector. So lentiviral vector, retroviral vector, and other adeno-associated vector. But if you come with a different lentiviral vector, we can apply our analytical method for the different gene of interest and for the different application. When a client comes to us for our platform method, we need just to verify the interference of the medium in which the vector is resuspended. But then the method is ready to go, and we can use as soon we start with the process development to analyze the different samples. For sure, we need to have the product specific that the client can transfer to us, or we can develop according to the client request.
It does sound like a solid foundation to have a platform for both LVV and AAV separately. I agree. Our next question is for Monica. I was hoping you could speak a little bit more about the flexibility that AGC Bio has in supporting clients with their process validation studies. I know in particular, PPQ readiness is a concern.
Yes, I think that AGC is considered can be considered a very flexible CDMO. The experience that I have is of greatest flexibility to accommodate clients' needs and client requests. We have had clients with more conservative or less conservative approaches when facing the PPQ preparation, and in all cases, we have done our best to support them in terms of the documentation preparation and respecting their timelines for activities completion. What we do is to be very transparent with them, and in case we see that the PPQ readiness is prepared in a very fast and quick timeline with not very huge data from stage 1. We make them aware of this, and we say them that this might be an issue in defining some of the acceptance criteria, as I mentioned before. But anyway, we every time do what the client asks us to do. So we do our best with the information and the data that we have in our hands. Of course, it is better if we have a long journey before we arrive at PPQ, since it gives us more time for preparation. But in the case there is no possibility to have a long journey and there is no budget to have a very long journey, we try to do our best with the material that we have in our hands. So yes, I can say we are very flexible. We should ask to our clients what they think about this, but feedback that we have had from them goes in this direction, that we are pretty much flexible.
Speaking of client timelines, I'm wondering how quickly can your AAV and LVV Bravo and Pronto platforms produce product for phase 1 activities?
I think we can say almost very quickly. Yes, we have good process knowledge since the development of these two platform processes. So when clients arrive using those, they already receive a bucket of information about the process, about process parameters understanding as well. And yes, we can start testing if the platform processes are fit for purpose of the client requests, and if that's work smoothly, we can say that in a few development runs at full scale, we can also move to clinical phase 1 manufacturing. Our platforms are developed using GFP vector, so it could also happen that if the clients bring a new vector, this might not behave exactly as the GFP vector, but generally speaking, it is quite quick.
I want to again thank our speakers, Monica and Francesca, and we thank you all for attending today's webinar. Goodbye and take care.